All right, this is Unit 2A. It's on the physiology of the renal system or urinary system. I'm going to start with a quick, brief review of the anatomy of the urinary system. I would remind you that the kidney has two major regions, a inner medulla and a outer cortex. Here you see the cortex, this outer area of the cortex. Don't forget it's 3 dimensions, so it's over the entire surface area. This is the inner area, the medulla. It's the boundary between the two, the cortex and the medulla, is delineated by the arcuate vein and arteries, which run here across this in this region right at the boundary. These are all arcuate veins and arteries. Urine is formed in the kidney, and we're going to go through that process later after we just quickly cover the anatomy. Approximately a quarter of the total cardiac output approximately 1/4 of cardiac output. Cardiac output is about 4.8 or 5 liters per minute usually. I remind you, cardiac output is determined by looking at the stroke volume times the heart rate. So stroke volume times heart rate normally would be about roughly 4.8 or 5 liters per minute. Approximately a quarter of that blood output would flow through the kidneys every minute. So the blood is constantly flowing through the kidney and undergoing a filtration process, which we'll describe soon enough. As I said, the arcuate artery delineates the boundary between the cortex and the medulla and the kidneys. The circulation in the kidney is unusual in that you have two capillary beds. One is a high pressure capillary bed formed by the glomerulus inside, which is a capillary tuft of capillaries inside the glomerular capsule, and then a second, and then the blood leaves the glomerulus. There are many glomeruli in the kidneys. Blood comes in via the afferent arteriole, goes into this capillary bed called the glomerulus, then leaves the glomerulus as in the efferent arteriole and forms a second capillary bed called the peritubular capillaries, and also another similar structure called the vasa recta. We'll show that in a little bit. The nephron itself is where the urine is being formed. Each kidney contains approximately 1 million nephrons. You have way in excess of what you need between the two kidneys, 2 million nephrons. You don't need that many, but they're so important that you have excess, and if they are damaged or destroyed, and slowly that excess reserve functional ability is destroyed until you get down to the threshold of the minimum amount needed. And once you get down there, well, you know, if that whatever process that's involved in the destruction of nephrons has not been detected earlier and slowed down or stopped, then you're in deep trouble once you start getting symptoms, things that you can feel and experience. Then you know, it's too late. Too much of your kidneys have been destroyed. The nephron, a single nephron itself, consists of the glomerulus, that capillary structure inside a glomerular capsule or Bowman's capsule, and also the renal tubules. The structure is shown here. There are actually two types of nephrons, cortical nephrons. Here, by the way, you see the corticomedullary junction, the junction or delineation boundary between the cortex, which is up here, and the medulla, which is down in this direction. So these are the arcuate arteries and veins that form that boundary that I mentioned. And there are cortical nephrons positioned in this way with a loop of Henle, this loop shown here. That loop of Henle actually normally goes a little deeper than is shown. It's maybe abbreviated here. But this is the normal structure. The glomerulus itself is up in the cortex always, and the loop of Henle dips down into the medulla. Again, I remind you that this is the medulla in that area, and this is the cortex up here. There are also juxtamedullary nephrons. These nephrons are located, the glomerulus of the nephron is located closer to the boundary between the cortex and the medulla, and the loop of Henle that they have dips deeper down into the medulla than the cortical nephron loops of Henle. And these are involved in production of a more concentrated, they have an ability to produce a more concentrated urine. The two capillary beds are shown here. There's the glomerulus, there's another glomerulus, and the second capillary bed is all of these capillaries that are surrounding the tubular structure here, shown here. These are the peritubular capillaries. If we look at the juxtamedullary nephron, what you have is down here, this network of peritubular capillaries has a particular name. It's called the vasa recta. Okay, the nephron, of course, is the glomerulus and the glomerular capsule or Bowman's capsule. Glomerular endothelium, that capillary bed endothelium, is fenestrated capillary epithelium, so it allows for a filtrate to be produced. That's what the kidneys do. They filter the blood plasma, and that filtrate is the fluid that is first formed and goes into the tubular system of the nephrons, and it is essentially a protein-free fluid which is very similar to plasma but completely protein-free. Passes from the blood into the glomerular capsule and then starts going down the tubular system. And that tubular system is shown here. There's the glomerular capsule with the glomerulus inside, and this is the tubular system. I'm outlining it here, shown in green. And it comes back and then goes to the collecting duct. And many nephrons send, quite a few nephrons will send their tubules down to join. Here at this point, you can see there's many nephrons feeding into one collecting duct. There are many collecting ducts, but there are many more nephrons. The structure of the tubules, I would remind you, starts with the glomerular capsule here and continues with the proximal convoluted tubule, this area here, and then the loop of Henle, which has a descending limb and then a ascending limb, and the tubular system then goes back up and comes very close to the glomerular capsule and glomerulus where it originated from, and now up here in this area in the cortex, it is called the distal convoluted tubule and goes to the collecting duct. Along the way, there are different cells that line the tubular system. I'm not going to get into that, but they're shown on the right here. The glomerulus itself is covered with a particular type of cell called podocytes. They form part of that filtration membrane, filtering the fluid that's going from the blood into the tubular system, forming the first fluid, which is called a filtrate. The collecting ducts, as I said, collect the fluid coming from many tubules of many nephrons. They empty into the collecting duct, and then the collecting duct is lined with cells that help maintain acid-base balance by changing the fluid, and also can help maintain water and salt balance by changing the fluid in the collecting ducts. Cortical nephrons that I talked about form 85% of the nephrons, and the juxtamedullary nephrons are about 15%. And here you see the two different types. The capillary bed, as I said, there are two capillary beds for every nephron. There's the glomerulus in here, and then the peritubular capillaries all surrounding all of the tubular structure all through there. And each glomerulus, each of that initial capillary bed is fed blood or receives blood from an afferent arteriole, and that feeds into feeds blood here, as you can see, coming into the glomerulus, and then it's drained by the efferent arteriole, leaving. This is all arterial blood, so it's under higher pressure, and you need that higher pressure actually, because the glomerulus is an unusual capillary bed in that it has a fairly high pressure, which is required for formation of the filtrate by pressure-driven filtration. Blood pressure, and this is, as I was saying, the blood pressure in the glomerulus is relatively high for a capillary bed. The arterioles feeding in and out are fairly high resistance vessels and can change the diameters. The afferent arteriole has a larger diameter than the efferent, and for that reason there's a reasonably high pressure inside the glomerulus because the inlet is larger than the outlet. Fluids and solutes get forced out of the blood through the entire length of the glomerulus by a process called filtration, right? The peritubular capillary beds, that second capillary bed, are much lower pressure, much more similar to the rest of the systemic capillaries, and quite porous capillaries that are adapted for absorption. And the peritubular capillaries are formed after, or are structures after, the efferent arterioles, and empty into the renal vein, the left and right renal veins, draining back to the heart. Vasa recta are specialized, long, very long, stretched out efferent arterioles leading to the capillary, the peritubular capillaries around the loop of Henle and the juxtaglomerular nephrons. Here you see these vasa recta coming down, and there are capillaries around this structure. Okay, there is a unique kind of resistance to blood flow within the afferent and efferent arterioles. As I told you, the afferent arteriole is a fairly high pressure arteriole. It has a wider diameter than the efferent, so blood coming in has an easier path in than going out, and therefore it maintains a fairly high pressure within the capillary bed of the glomerular capillary bed. Blood pressure will decline from, in the renal arteries, it's at 95 and goes down dramatically to the renal veins, but in between you have these capillary beds. The first one, glomerulus, is a fairly high pressure, under fairly high pressure to facilitate the filtration process. Resistance to flow by vasodilation, by vasoconstriction, will protect the glomerulus from, and vasodilation, any changes in resistance to flow by changes in the diameter of the afferent arterioles protects the glomerulus from any fluctuations in systemic blood pressure. Your systemic blood pressure is changing all the time as you stand up and lie down and run or exercise or bend over or contract your muscle. There's always these fluctuations going on, but the glomerulus itself is protected, and a fairly steady maintained pressure is always present in the glomerulus. And that's maintained by alterations in the diameter of the afferent arterioles. Both constriction and dilation can maintain that pressure. Resistance in the efferent arterioles helps to reinforce the needed high glomerular pressure. So if glomerular pressure is too low, you get a little constriction downstream, and you get constriction in the downstream efferent arterioles, and that raises the pressure. If you get any dilation, that'll lower pressure if it's too high in the glomerulus. It helps to maintain proper hydrostatic pressure in the peritubular capillaries and prevents it from getting too high. So it can reinforce or reduce pressure to help maintain that glomerular pressure at adequate levels. Here you see an afferent arteriole coming in, and then you see the glomerulus in cross section, the thin little capillaries of the glomerulus, and you see the red cells going through. Remember, in a capillary, red cells essentially are going through single file. They're very, very, very thin, so this is an artist's drawing. There's many, many more. There would normally be, if you did a photomicrograph, you would see many more cross sections of the glomerulus. There's the outflow from the glomerulus, the efferent arteriole here. Is showing the flow outwards. This is flow inwards, the afferent arteriole. Remember, the filtrate, the first fluid formed by the filtration process, goes into the glomerular capsule and then flows down the proximal convoluted tubule. And that's shown here, and it will go through that and then into the loop of Henle and then back up into the distal convoluted tubule. And I told you that the distal convoluted tubule always comes back near the glomerular capsule where it originated. And here you see that distal convoluted tubule in cross section. In the wall of the distal convoluted tubule, right here, where it comes back near the nephron where it originated from, is a structure called the macula densa. Our macula densa cells, which form macula densa cells here in the wall, which form part of what's called the juxtaglomerular apparatus. Juxtaglomerular apparatus is made up of macula densa cells in the wall of the distal convoluted tubule, the juxtaglomerular cells, which are found surrounding mostly the afferent, but also a little around the efferent arteriole, and also mesangial cells, these gray cells shown here all throughout. So that's the juxtaglomerular apparatus. What it does is acts as a mechanoreceptor, looking at the juxtaglomerular cells, at least, act as mechanoreceptors to respond to stretching of the tubules, and the juxtaglomerular cells contain a hormone called renin in granules inside those cells. It's not only made up of juxtaglomerular cells, but also the macula densa cells and the mesangial cells. The macula densa cells act as chemoreceptors or osmoreceptors. They monitor the quality of the fluid in the distal convoluted tubule. And I'll talk later about the physiological function of the juxtaglomerular apparatus. Mesangial cells spread throughout that juxtaglomerular apparatus have phagocytic and contractile properties. They help influence capillary filtration, but I'm not going to get into them too much. So here again you see the juxtaglomerular apparatus made up of these three types of cells. It's the just glomerular cells that contain renin, but macular density cells also have an important function. The filtration membrane of the glomerulus, the cells between the blood and the interior space of the glomerular capsule, those membranes and whatever connective tissue is there form a filter. So the blood plasma is pushed across that filter and enters, and the first fluid formed that will eventually lead to becoming urine, will eventually become urine. It's called a filtrate, and it's pushed into the glomerular capsule. The filter itself is composed of the fenestrated endothelial cells that line the capillaries. They pave the inner surface of the capillaries, and the membrane of the glomerular capsule is formed by cells around those capillaries. They're called podocytes. I'll show you that in a diagram in a minute. And then the basement membrane in between those layers. And here you see it. This is the inside the glomerular capillary. So there's plasma, and the direction of the filtration process, the fluid being formed into filtrate, is in this way. So this is the inner space here. This area, all this area, these areas are the inner space of the glomerular capsule. The endothelium of the endothelial cells are shown here in beige. I'll highlight it a little bit here. These cells are the endothelial cells. There are fenestrations or windows in between. It's a fenestrated capillary bed. There's the basement membrane and the podocyte. Podocytes have these foot processes that extend over the surface of the glomerular capillary. I'll show you on the next diagram what that looks like. So that's the filtration membrane. Here you see a fenestrated capillary, part of the glomerulus. You can see the little holes in it, the fenestrations, and you see the podocyte on the surface of that, forming with these filtration slits where filtration actually occurs. Okay, we'll finish off this. This is all review of anatomy that you should have covered last semester, but I wanted to, remind you of it. This is on the left. It should be hopefully obvious to you that is the male ureters coming from each kidney into the bladder that stores the urine and then empties it out through the urethra. And it goes out here, the base of the penis, and obviously, hopefully to all of you, on the left side of the slide is the male, the end of the male urinary tract, and on the right is the female, and the two ureters again going into the bladder and then feeding out through the urethra to the outside. For, again, hopefully to you, obvious reasons, females are more susceptible to urinary tract infections. Shorter urethra, easier entry of bacteria into the bladder to cause a urinary tract infection. So females get them more often than males. Males also get them. This imaginary area forming into a triangle, which is formed by the opening of these ureters here from each kidney, open into the bladder down here at the base, and then the urethra opens here, and the imaginary triangular area is called the trigone. Again, the two openings, 1, 2 of the ureter into the bladder, and then the urethral opening forms the third the third corner of the triangular structure, anatomical structure called, or area called, the trigone. Okay, so let's move on. Okay, well, in general, the kidney filters approximately 200 liters of blood daily. That's a rough estimate. So you obviously don't contain, the average person doesn't contain 200 liters. It's going round and round constantly through the kidneys, as I said earlier, and being filtered, the plasma part of it. Now, in that filtration process, what happens is that some toxins and certainly metabolic waste molecules and excess ions are excreted by the body into the urine, and overall also any excess water. Everything has to go out dissolved into water, and you put out more or less water depending upon your body's needs. It's regulated carefully. So the volume and the chemical makeup of the blood is regulated through the action of the kidneys mostly. They maintain a proper balance between water and salts and acids and bases. Other functions include things that we won't, some of which we won't talk too much about, some of which we will. The kidneys also can carry out gluconeogenesis during periods of prolonged fasting, so they can create new glucose from non-carbohydrate sources. They are also responsible for renin production, which I mentioned earlier, and that helps to regulate blood pressure. I'll talk about that more soon enough. They are able to produce erythropoietin, which is a hormone that stimulates production of red blood cells in the bone marrow, in the red bone marrow. And they act on vitamin D to activate it. So they're quite important in that production of that vitamin. Well, in terms of the mechanisms of formation of urine, the kidneys are constantly filtering the plasma as it moves through the kidneys many times per day. The very first fluid formed, as I told you, the very first fluid that forms through the process of kidney filtration of the plasma through the glomerulus into the through the glomerular wall into the glomerular capsule is called the filtrate, and that filtrate contains the exact same components as the plasma minus any proteins. So it's everything except for proteins in terms of what it looks like. It's very similar to the plasma minus proteins. But that's actually still, you know, the amount of filtrate that's being formed is quite large compared to what actually comes out as the final end product of urine. So the filtrate will undergo a lot of processing as it moves through the tubular system until it finally leaves as urine. This is a diagram showing the glomerulus, blood coming into the glomerular capsule to form the glomerulus, that first capillary bed, comes in via the afferent arteriole, goes into the glomerulus, and undergoes the process of filtration. This would have been better as an A instead of a 1, glomerular filtration. And that's where the fluid, the filtrate, enters into the tubular system and then progresses through the proximal convoluted tubule into the loop of Henle, and then to the distal convoluted tubule, and finally into the collecting ducts and down the ureters to the bladder, from where it gets excreted through the urethra as urine. So this diagram actually shows it's a stylized diagram depicting all of the tubular system all the way down to going out as urine. As it goes through, the filtrate that forms in the glomeruli, that was formed from glomerular filtration, undergoes two processes: reabsorption and secretion. And reabsorption and secretion are carried out together with the peritubular capillaries, which receive reabsorbed fluids or secrete materials in insoluble form into the filtrate. So these are the three processes that are going on during the formation of urine: filtration, reabsorption, and secretion. We're going to go through each one and look at, these are the three major processes, and we'll go through each one and look at their regulation. Glomerular filtration involves the basic principles of fluid dynamics, same principles that were operating in the blood capillaries also operate in the formation of filtrate in the glomerulus. Unlike other capillary beds, the glomerulus is more efficient in terms of fluids that move across, that get filtered across into the tissue, into the tubular space. The membrane is significantly more permeable than what you find in capillary. Remember, in blood capillaries, as the blood goes into a capillary, most capillaries in the systemic circulation in tissues, some of that fluid comes out and goes into the interstitial space, and then most of it gets pulled back into the capillary, but some stays and works its way into the lymphatics. In terms of the glomerulus, filtration is significantly greater. A lot more fluid goes across because the glomerular filtration membrane is much more permeable. It's a fenestrated capillary, and also because glomerular blood pressure is very high. There's no other capillary bed that has as high blood pressure. So overall, in fact, it has a much higher net filtration pressure. Blood plasma proteins do not get filtered. They don't exit the bloodstream at all. And they in fact help maintain what's called colloid osmotic pressure of the blood, or you might also see it called oncotic pressure. This refers to the ability of proteins within the bloodstream inside the glomerular capillaries to pull fluids in and help move fluids, prevent excess amounts of fluids from leaving. So those plasma proteins, and one of the most significant ones is, of course, albumin. Albumin forms about 50% of the proteins in the blood plasma, and is quite a large molecule and cannot go out of the glomerulus into the tubular space, into the glomerular capsule space. And so what you have is a number of forces acting, and together they form the net filtration pressure overall, the pressure responsible for filtrate formation. So the forces that are operating include the hydrostatic pressure in the glomerulus, which is around and is kept fairly steady at about 55 millimeters of mercury. So that's abbreviated hydrostatic pressure of the glomerulus, G, subscript G. So that's shown here in these purple arrows, showing that there is blood pressure pushing filtrate out. Then you have the colloid osmotic pressure of the blood, or the oncotic pressure of the blood, due to large protein molecules that can't escape and can pull in fluids or retain fluid in the glomerulus, and that's abbreviated OP, osmotic pressure of the glomerulus, G, subscript G. And then, so that's an inward force, and that's shown in these light brown arrows. And then you have the capsular hydrostatic pressure, HPC for capsule. That's, again, an inward force. Now, when you add all of these up, and the numbers may vary depending upon what source you look at, which book or file or figure or whatever you see, these add up to an outward, they may vary, but in the end you have approximately an outward net filtration pressure, net outward pressure, net filtration pressure of about 10 millimeters of mercury. So there's an ongoing outward flow. In general, there is an ongoing outward flow. The blood glomerular hydrostatic pressure is steady. The blood colloid osmotic pressure is pretty steady. Capsular hydrostatic pressure is reasonably steady, and you have overall an outward flow. There is missing, of course, you might notice, osmotic or a colloid osmotic pressure of the in the capsular space. That would be the osmotic pressure of the filtrate fluid in this space. It doesn't have any significant colloid osmotic pressure because it's free of proteins. So that's the net filtration pressure. Overall, the equation is, and the net filtration pressure is equal to the glomerular hydrostatic pressure minus the other, that's an outward force. Okay, so let's say up is outward and down is inward. So a net, the net filtration pressure is the hydrostatic pressure in the glomerulus, so net outward force minus the sum of the inward forces. And by inward, I mean from the capsular space pulling fluid into the glomerulus. So the sum of those two, and those two are, of course, the colloid osmotic pressure, or also called oncotic pressure of the glomerulus, and the capsular hydrostatic pressure. These two, the sum of these two subtracted from the glomerular hydrostatic pressure is equal to the net filtration pressure. Okay, I think what we'll do is stop here. Slide 32 of Unit 2A, and that'll be the end of Unit 2A Part 1.